Let us get started. Today is Tuesday, November 23rd, 2021. It is always a little scary and exciting, both exciting and scary when we can get the entire rest of the schedule on the board. And yet that is where we are at. We are rapidly approaching the end. We have one more organ system. We have 4 more lectures to go and we are done. As you can see, it's still going to be busy, but spread out time. We meet today, and then we have yet another holiday on Thursday, Turkey Day, so no lecture on that day. Tuesday, we do come back. There's nothing due on that day, which of course, as you know, means it's probably a good day for a daily quiz. Thursday, you have a labster. Thursday the 2nd, you have a labster due. He's on, I know it's called endocrinology, but it really has to do with the reproductive system. So we'll be doing that. There's a second labster on Mendel. We're going to sneak a little genetics in at the end of this semester. And then also one more unit review, unit 27. And then it's exams. Thursday the 7th is your last lab and lecture exam. Again, we have just the way that the holidays fell, because of the way the holidays fell, another Thursday exam day. That means that, again, Tuesday, you will be getting some new material that you will be responsible for 2 days earlier. Oh, I have those dates wrong, don't I? What is it? Is it the 9th is the Thursday the 9th. So that means your weekend, the 5th and the 6th, are your last big weekend to study for the exam. So you will do that. We'll get a little bit of new information on the 7th. We'll try to front load it as much as possible, so it's not too much. But remember, you do have the slides, you do have the outlines, you do have the other materials. And that day we'll be talking primarily about development. And if you looked at the chapter, there's a huge amount of information. You were just going to be responsible for the stuff we cover in class, so it won't be too bad that way. Then a nice long 5-day weekend, and you come back for your 100-point cumulative final exam. Again, it is 100 questions, 100 points. You'll have about 2 hours to complete that in, and that is, remember, counts as a grade on its own, but also can be used to replace your lowest lecture score. So if you stumbled somewhere along the way, this is a perfect opportunity for a nice big swing to improve your grade. And that's it. Then on the 15th, you get to sleep. All right, questions on any of that? Excellent. Nothing like I love more than my stunned silences. Yes, absolutely. It is a 100-point multiple choice. All questions are multiple choice questions. So again, there's a huge advantage to that. You don't have to pull the information out of the ether. You just have to recognize the correct answer. I do not pull the questions just from the questions that have been on the previous exams. 1 or 2 may fall in there, but it's, you know, because I don't care about that. But it covers all the material. So yes, so you just have to recognize the information. Yep, lab stuff and lecture stuff, everything we've covered in this class. But so no problem, Max. All right. Any other questions? Oh, great questions. No, again, I'm not going to reopen the previous exams. You've had the opportunity to look at those as we've gone through. And honestly, I think looking at the previous exams is a horrible way to study for the final, because you got to remember that in each of these units, what was on the test was a small portion of the material that we covered in that section. And so the final isn't made up of the stuff we did on the exams. It's made of the stuff that we did in the class. So just because a concept wasn't on the second lab exam doesn't mean that it's not going to be on this exam. So, or again, the example I use that I like the best because it's easiest to understand is if you think back to 430 when we did the muscle contraction. We broke it into 3 parts: communication at the neuromuscular junction, excitation, contraction coupling, and the contractile cycle. And in my class, 2 of those were essay questions on the exam. So if the neuromuscular junction and excitation, contraction coupling were your 2 essay questions, then you didn't have an essay question on the contractile cycle on the exam. Does that mean the contractile cycle is not going to be on the final? No, of course not. And so again, using your exams to study is a very poor way to study for the exam. Best to go back and look big picture at the material that you've covered for that. Let's see what else we got here. There may be some labeled pictures. Again, I may have a heart, you know, with an arrow pointing at the superior vena cava or something like that, and you need to recognize it. But again, it'll still be a multiple choice question. So it'd be probably a labeled picture with, you know, identify the structure labeled A or something like that. There could be some of those types of things. I wouldn't say it's a huge component of the exam, but there will be some of that on there. Let's see what else we got here. Yeah, totally. Ruby, that's not a problem at all. And yes, because it is not a lab exam where we have, you know, very dense pictures and a lot of them, it will be presented all at one time. So you will have the opportunity to go through it. And again, because the nature of the online format, it'll for the most part be in order, section by section by section. That is not the case normally on my finals. My finals just a mix of all the information. So that's another advantage. You'll get all the cardiovascular system stuff and then all the respiratory stuff and then all the urinary stuff and so on and so forth that way. So that actually can help a little bit as you're going through that. But yes, it will be presented all at once because it's multiple choice questions. So it'd be more like the lecture exam that way. Awesome questions. Any others? All righty. With that, let's dive into the reproductive system. Last organ system. I say system singular, but as we've kind of talked about in the past, it's really an organ system plural, systems. Obviously, the male and the female reproductive systems have to work together to be able to produce offspring, but they are also dramatically different from each other as well. So while they have very similar goals, the ways they go about doing it are very, very similar, but also different as well. And so it's almost like learning two organ systems. However, of course, as we mentioned, there are some things that they do share in common. For starters, they're the only organ system really that is not continually functioning. You know, luckily your cardiovascular system has been working since before you were born, right? And it doesn't get Sundays off or it doesn't get a late start on Wednesday or anything like that. It is a continuously functioning system. However, our reproductive system is not functional when we are born. It matures as we do. And then for females, actually, that functioning stops. So it is not a continuous system. So that makes it very unique. Of course, what are the primary sex organs?
Testes and ovaries.
Yeah, exactly. The gonads, what we would collectively call the gonads, the testes in males and the ovaries in females. I always love that pregnant pause when I ask that question because everybody's nervous about saying penis and vagina. But remember, this is an anatomy and physiology class. We are allowed to say penis and vagina. They're just not the right answers. Gonads are the right answers. They are the ones that are the primary organs, whereas again, obviously the penis and the vagina are important copulatory organs, but they wouldn't be what I call the primary ones, because the primary ones, as we've talked about, are the ones that are responsible for the reproductive functions. As we mentioned, our gonads make the puzzle pieces, or at least that's how I explained it to Big when she was, 3 years old and asking about, how mommy and daddy made babies. Mommy and daddy make the puzzle pieces, put the puzzle pieces together and made you, right? And that's the gonads, that's the sex cells, the ova or ovum singular in females and spermatozoon singular in males. Plural, spermatozoa and ova. Females, again, we see that disparity, get the honor and the privilege of also housing and nurturing those puzzle pieces for 40 weeks until it is ripe enough to pop. and then they get to live on their own after that. Of course, remember, as we talked about, this isn't the first time we've talked about the gonads. Remember, we talked about the gonads in their relationship to producing the sex hormones. And as we talked about, the function of those play an important role in the growth and development of many organs and tissues, that maturity, that converting a boy into a man, that girl into a woman. Right, the development of the reproductive organs, making them copulatory ready and ready to reproduce. And we even talked about, remember how, for instance, some of the gonadotropins that are produced by the adrenal gland, as well as the testes and the ovaries, play an important role in libido, aggression, sex drive, things along those lines as well. All right, questions on all of that? All right, I think I used this analogy before in this class, but I'll use it again just in case. As I mentioned, the reproductive systems for the males and the females are similar in much the same way that a 1968 Volkswagen Bug and a 2022 Lexus are similar. They're both cars, they both have engines, they both get you from point A to point B. But obviously, if you didn't know anything about cars, which of those two would you want to study first to understand how a car functions? Yeah, the Volkswagen Bug, absolutely. Right? They both basically do the same thing, but that Lexus has a lot more bells and whistles. But as you're trying to understand this, it's best to work with the basic model first. And that's what we're going to do here as well. Males are the 69 Bugs and females are the 2022 Lexuses, like Lexi, I don't know, whatever the plural of Lexus is. The point is the male reproductive system is far, far simpler. So we'll start with that as our basic model. And then from there, once we have an understanding of that, we'll go on to the more complicated version, which is the female version. Here we see the male reproductive system. Again, we have that primary organ, the testes. And then essentially what we have is a closed duct system. By that, what I mean is from when that sperm is first produced in the testis, the ductwork system that carries it and ultimately expulses it from the male body is all connected. So from the testis, there's only one place that it can go. From the epididymis, there's only one place it can go. It is a completely closed system. And I've made a point of emphasizing that 3 times now, because as we'll learn when we see the female's ductwork system, it is not a closed system. It is an open system. This is one of the reasons why sexually transmitted diseases are much more dangerous for females than they are for males. If some type of disorder gets into the reproductive tract of a male, it stays in the reproductive system. Whereas the female's reproductive system is actually open to the entire abdominal pelvic cavity. So a sexually transmitted disease that gets into the female reproductive tract can potentially affect any organ in the abdominal pelvic cavity. It can affect the digestive organs and all sorts of other stuff inside of there as well. Those ductwork structures in the male include the structures, the epididymis, the ductus deferens, the ejaculatory duct, and our friend that we talked about from the last organ system, the urethra, which as we can see here again has 3 components to it as well. Remember, there is the prostatic urethra that goes through the prostate, membranous urethra that goes through the urogenital diaphragm, and a lot of people got wrong on their lab exam, and the spongy urethra that goes through the penis. So if you struggled with that membranous urethra, well, I got to make myself a note to release the exams. Someone remind me at the end of class that if I forget to mention it again at the end of the class, that your exams have been graded and I want to release them for you so you can see this last exam before the break. And so again, you'll get a chance to use and learn and understand those 3 distinct regions of the urethra for this as well. Lastly, there is the external genitalia. There is the penis and the scrotum sac, and also there are accessory organs, the glands. There is the single prostate, the paired bulbourethral glands, and even though this picture only shows one of them, there are posteriorly behind the bladder here 2 seminal vesicles. So the seminal vesicles are paired, the bulbourethral glands are paired, and the prostate is singular. And then, like I said, the external genitalia as well. All right. Questions on that? All right, we're gonna have a lot to say about the testis, but before we get there, let's talk about the scrotal sac. And again, here we saw it in this kind of longitudinal view. Here we see all of these structures in a sagittal view. Like I said, we're gonna spend a lot of time talking about the testis, but let's talk about where they are housed. There is an extension of the skin and the superficial fascia out of the abdominal pelvic cavity to form the structure known as the scrotum. And it is beneath the root of the penis. It contains the paired testicles, and each one of them have their own individual compartment within that scrotal sac. So they're separated compartments. They're just not just like a couple of quarters rolling around in your change purse. The scrotum, and actually let's draw a very simplistic version of this, because then we get some fun with vocabulary. And not surprisingly, genetics and reproduction go very much hand in hand. And the scrotum is a homologous structure to the labia majora of the females. What does that mean, homologous structures? Anyone know?
Like very similar, or can like almost parallel?
I like beyond similar, I like parallel a lot better. Basically, when we all started as a ball of cells, as those cells divided, you had this cluster of cells. And this specific cluster of cells can do one of two things. In females, this structure of cells become the labia majora. Basically, the outer folds of the vestibule for a female that extend down and protect the orifices of the urinary and reproductive systems. In males, that exact same bundle of cells, basically, instead of just forming sleeves on the side, extend out and actually connect in the middle. So basically, homologous structure basically means that they are made from the exact same set of cells. And in males, they do one thing, and in females, they do another. So basically, it is just the extended labia in the females that form the scrotum. You can actually see this homologous structure and this development, because at the midline, where basically the two folds come together, is a raised ridge known as the raphe. For those of you who have scrotums, this is something that you can observe for yourself. Along the midline of the body, there is a raised ridge on the scrotal sac. If you do not happen to have your own, feel free to borrow one in the household that you live to take to observe that. And if you don't happen to have a scrotum in the household that you can observe, I'm certain that if you go to any bar, assuming that you are properly vaccinated, and you ask any gentleman who's standing at the bar there to observe his raphe, he would be happy to show it to you. So it's a perfect opportunity to get a chance to meet people and do some anatomy and physiology studying. So you have that structure on the midline that is known as the raphe, where those two come together from the side. What is the point of the scrotum, other than obviously to win $10,000 on America's Funniest Home Videos? What is the point of having the testes in the scrotum? When you speak with some biologists, they believe that our primary function as an organism is to reproduce, to produce more of our species. And so if it's that incredibly important of a function, then why would you take the structures responsible for that and house them in such an unprotected place outside the body? Exactly. Too high for what?
Sperm production?
Exactly. You guys have the right idea, although let's be careful. You have the right idea. It's there we go. Maximilian's got it. A male is perfectly capable of producing sperm, even if the temperature of his testes is too high. However, the sperm will not be viable. They will not be capable of fertilizing an egg. So basically what happens is we have to maintain a lower temp in the testes to produce viable sperm. Or you could say it the way Maximilian did, for sperm to mature. So the maturation of the spermatozoa requires a temperature about 3 degrees lower than core body temperature. Now, the testes actually start in the abdominal pelvic cavity. Again, obviously, they're the homologous structures to the ovaries in females, and like the ovaries in females, they start in the abdominal pelvic cavity. And then just about the time of birth, they descend into the scrotal sac. And so at the time of birth, they are already in the scrotum. Most of the time. There are occasions where one or even both of the testes do not descend prior to birth. And for the longest time, that was thought to be okay. Right? After all, they're not going to need them till they're at least, well, they should need them till they graduate from college, but it's not going to really become functional till they're somewhere between, 14 and 16 years of age. So if it takes a year or two for them to descend, right, it's not a big deal. If maybe by the first birthday they haven't descended, then they would do the surgery then. So they didn't think it was that big of a deal. However, what they have since found is that if the testes do not descend, if they stay in the abdominal pelvic cavity for a prolonged period of time, it dramatically increases the likelihood of testicular cancer. So now typically if a baby is born with one or both of the testes having failed to descend, usually within the first month or two, they will surgically descend it into the scrotal sac just because of that increased risk of testicular cancer from that. So it's something that they pay much more attention to. Obviously, they're not going to need it for years and years and years, but because of the risk of testicular cancer, it's something that they do much quicker now than they used to do before. As I mentioned, this is an extension of the abdominal pelvic cavity, and so not surprisingly, we have an extension of the peritoneum. Just like the peritoneum in the abdominal pelvic cavity, it is going to have, the testes is going to be surrounded by not one, but 2 layers of the peritoneum. And of course, we can't get away with just simply using a simple term like peritoneum because anatomists hate us. So of course, we are going to have the visceral that is surrounding the testis and the parietal that lines the space around it. And instead of the peritoneum, it is known as the tunica vaginalis. The tunica vaginalis is contained within a structure known as the spermatochord. So the spermatochord is basically this fibrous connective tissue structure that wraps around the testis, wraps around the blood vessels, wraps around some muscle, as we can see here, providing that protection. And as you can also see, the spermatochord passes through the inguinal canal. If you remember way back in 430, we learned about the inguinal ligament. Remember the inguinal ligament basically goes from the superior anterior iliac spine to the pubic tubercle. It is a ligament, bone to bone. It is an attachment point for the abdominal muscles and several other muscles, and it forms within there a space known as the inguinal canal. In females, they also have an inguinal ligament, and they actually also have an inguinal canal. But in females, it is a potential space. What do I mean by potential space?
They can or can't have it.
Not necessarily, although I like what you're thinking of there. Let's think of it this way. If I take my two hands and I mash them together, would you say that there is anything between my hands?
Not much.
Yeah, nothing. I would say there's nothing between my hands, right? However, have my hands fused together into a single structure?
No.
No, of course not. So notice there is the potential for a space there. I could make a space in there that I could put something in if I wanted, but as long as it remains flat, it's basically closed and just a potential space. And that's basically what it is with females. Basically, it is a potential space. So in males, it's an actual space that spermatocord passes through. In females, it's just a potential space. And make a point of emphasizing this because this opening is an opening to the abdominal pelvic cavity. And as we talked about way back in 430 when we were learning about the muscular system, if the pressure gets too high in the abdominal pelvic cavity, that pressure may try to find a weakness where it can force itself out. And so what can actually happen is you can get a bulging of fluid or a piece of omentum or even a small piece of an organ like the small or large intestine can push its way out that weak spot in the abdominal pelvic cavity. And what do we call that type of condition?
A hernia?
A hernia, absolutely. For males, it is much, let me say it this way. One of those sites of the weakness is here in the inguinal canal. However, do males and females, are they equally likely to get an inguinal hernia? No, it is very, very rare in females because it is a potential space. Whereas because it is an actual opening in males, an inguinal hernia is much more common in this location. Now, there is a second location that both males and females have that is equally likely to have a hernia. And what is that second location? The umbilical region. Absolutely. Excellent. Thank you, Eric. Absolutely. Everybody has a belly button, and that is a weak spot. So both males and females are equally likely to get an umbilical hernia, whereas males are much, much, much more likely to get an inguinal hernia. All right. Questions on that? All right, so let's go back to our picture here. Here in our picture, let's cheat, see if I can bring this whole thing down. Did a horrible job of this. Probably should have just redrawn it. That's all right. So, as we just mentioned, we have the testes. Nope. That's what I want. Each in their own individual compartment. No. Within the scrotal sac, and I'm going to cheat, put them a little bit more towards the middle so I have a little bit more room. Excellent. As we just mentioned, there is a visceral tunica vaginalis that wraps around the outer surface, and then a parietal tunica vaginalis that wraps around it as well. This is all contained within our fibrous spermatocord that carries it out and is the passageway for our blood vessels, arteries, veins, and nerves. Now, as we mentioned, having it descended in the scrotal sac helps to maintain a lower temperature. But as we also mentioned, we need it to be 3 degrees lower than a typical normal core body temperature. However, the environment that our body is in, the type of clothes, the layers of clothes, whether we're in a hot tub or whether we're in a cold bath or any of those types of things, can change from moment to moment. So just having it outside of the body isn't sufficient. So to help to regulate temperature, there are 3 important structures that are going to assist in this. 2 of them are muscles. The first is what is known as the dartus muscle. The dartus muscle lines the scrotal sac. And because it lines the inner surface of the scrotal sac, it's basically a curved muscle. And what do we know about curved muscles when they contract?
Become flat.
Exactly, they become flat. And as they become flat, basically that shortens the size of the scrotal sac. When they relax, it curves more and the scrotal sac descends. So basically this can change the shape of the scrotal sac, which can bring the testes closer to the body for warmth or further away from the body. So in this way, we can basically change the shape or the size, if you want to think of it that way, of the scrotum. Now, of course, this is smooth muscle, right? Can a male voluntarily change the shape of his scrotal sac?
No.
Of course not. So this is autonomically controlled. Exactly. However, there is a second muscle as well. So that was our dartus muscle. The second muscle is what is known as the cremaster muscle. The cremaster muscle actually attaches to the pelvis, and it basically wraps around the individual testis. So basically attaches from the pelvis around the individual testis. So not only can we change the shape of the scrotum, but the cremaster muscle actually can elevate and descend the testis to be able to bring it up or bring it down, again, closer to the body for warmth, further from the body if it's too hot, and move it that way. And this happens to be skeletal muscle. Now, does that mean that a male has to be consciously aware all the time of what the temperature of his scrotum is so that he is able to manually adjust the length of the cremaster muscle to put the testis in exactly the right place? No, of course not. But that's not any different from the fact that while you've been sitting here listening to me for the past 35 minutes, have you been consciously thinking about how often and how frequently and how hard you should blink while you've been listening to me talk? Has everybody blinked at least once during my lecture for the past 35 minutes? Yeah, and did anybody consciously think about it? No, absolutely not. And so it's the same thing here. Obviously, this is controlled via reflexes, right? However, it is skeletal muscle. So can a male voluntarily move it? Can you voluntarily close your eye if you want to?
Yeah.
Absolutely. So when you meet that male at the bar, not only ask to see him, to see his raphe, but you can also ask him to flex his cremaster muscle for you, and be sure to be impressed when he does. All right. Questions on that? All right. Notice I said there were 3 important structures that help us to regulate temperature. 2 of them are these muscles. The third is something we're familiar with from the last section. A countercurrent mechanism. Someone remind me again what a countercurrent mechanism is?
Two fluids going in opposite directions, almost touching each other through like a tube. And they
Okay.
allow for certain things such as heat to pass through.
Exactly. And that's exactly what we have here. If you remember way back when we were talking about the urinary system, and I first introduced the idea of a countercurrent mechanism, we talked about it in terms of a heat sink. Something that is capable of drawing heat away. Notice we've talked about how the testes need to be maintained at a temp that is about 3 degrees cooler than body temperature. But they're also needing to make sperm. And making sperm is a highly metabolic active process. And if you've got a highly metabolically active process, what do you need? These aren't trick questions. Something is very metabolically active. What does that structure need?
ATP.
It needs to make ATP. And what are the 2 things you need to make ATP efficiently?
Glucose and oxygen.
Oxygen and glucose, absolutely. And what is really good at bringing oxygen and glucose to places?
Blood.
Blood, right? So excellent. Not surprisingly, we have a large artery that carries blood to the testis. If you remember way back at the beginning of this class, when we were learning about the cardiovascular system, we mentioned how the testicular artery was larger than the ovarian artery because of the more metabolically active. But what else did we learn about blood when we were way back at the beginning of 431?
It brings heat.
Yeah, it carries heat. Remember, blood is 2 to 3 degrees warmer than your core body temperature. So it doesn't do us any good to have the scrotum, to have these muscles and all of this, if we have this really hot blood that is traveling to our testes and warming our testes up. Luckily for us, we have a heat sink. And that heat sink comes in the form of an elaborate network of veins that wraps around the artery. This ultimately comes together to form the testicular vein. But this elaborate mesh-like portion of the testicular vein is known as the pampiniform plexus. And so basically what happens is the pampiniform plexus contains cool blood. And so while this cool blood moves up and across the testicular artery, it basically draws the heat out of the blood. This warms the blood. So now we have this nice warm blood going back to the heart. And it cools the blood so that cool blood reaches the testis. So in this fashion, we have this heat sink, which basically draws the heat out. And then, of course, this cool blood goes back through the pampiniform plexus, drawing the heat out of the warm blood that's in the artery, and the process continues. Notice I've done a truly amazing job of drawing these structures here, but let's look actually at the pretty pictures from your textbook. Again, we have the dartus, which is smooth muscle, wrinkles and shortens the scrotal sac, or loosens and descends the scrotal sac. Our cremaster muscle, which elevates the testis. And then that pampiniform plexus, that is that elaborate testicular structure. So here we see the pretty picture up close, and we see these three structures. So again, notice lining the scrotal sac, we have that dartus muscle. That again is going to change the shape of the scrotum. We have the cremaster muscle anchoring to the pelvis, wrapping around each individual testis to elevate or descend it. And then here we see this really, really beautiful image of, again, notice we can see the testicular arteries coming in here, and this big elaborate mesh-like network, this pampiniform plexus that wraps around, carrying the blood back up, drawing the heat out of the testicular arteries. So not just having them outside the body, but also having these three additional structures, specialized structures, help us to maintain appropriate temperature so that the sperm produced are going to be viable. They will mature and they will be capable of fertilizing an egg. No, great question. No, remember, what happens is as the blood comes down the artery, it is cooled. So when it reaches the testis, its heat has been drawn out of it. So it is cool in temperature. Then as it goes back in the pampiniform plexus in the vein, it draws the heat out of the blood that is coming to the testis and warms back up again. So the blood has heat drained out of it as it goes down to the testis. And then as that blood goes back to the testis, it draws the heat out of the new blood to warm itself back up. Excellent. All right. Questions on this? So there you go. It's all about temperature regulation. And like I said, winning $10,000 on America's Funniest Home Videos. All right. Here we get another great view of that tunica vaginalis we were just talking about. Again, the tunica vaginalis is that extension of the peritoneum. Notice we have the outer layer where it is in contact with the outer surface or forming the outer surface of the testis. That would be the visceral layer. And then we have the parietal layer, and of course, a little bit of serous fluid in between them. But the bulk of the testis is made up of this white fibrous material known as the tunica albuginea. Albuginea, like albino, meaning whitish tissue. So if we again go back to here, and let's go ahead and use a different page. I don't like that. So here's our testis, not drawn to scale. Again, on the outer surface, it is going to have that visceral tunica vaginalis. It is surrounded by the parietal tunica vaginalis with a little bit of a cavity containing serous fluid inside. But the bulk of it is this fibrous white mass known as the tunica albuginea. And not only does it form the outer mass of the testis, but there are extensions of this tunica albuginea that extend out deep into the testis. These extensions of the, that's not an arrow. These extensions of the tunica albuginea form septa, or since I'm pointing at just one, a septum, which we know is, of course, a dividing structure, and divide the testis into somewhere between 250 and 300 individual lobules. Tiny compartmentalized spaces. So if we again go back to our illustration, we can see a representative of that as well. They don't extend it as much, but you can see how the tubules contained inside are compartmentalized by these extensions of the tunica albuginea, dividing it into these individual lobules, about 250 to 300. Each of these 250 to 300 individual spaces, these lobules, each contains somewhere between 1 and 4 seminiferous tubules. And the seminiferous tubules are the structures that are both responsible for, well, they're responsible primarily for making the spermatozoa, but we'll see there's some other important functions to it as well. So again, if we go back to our picture, each one of these contains anywhere between 1 and 4 seminiferous tubules. Each seminiferous tubule is about 3 feet long. They are surrounded by myoid cells. We'll see that when we look at the histology in a minute. With a name like myoid cell, what do you think myoid refers to?
Muscle?
Yeah, it is a very thin layer of smooth muscle cells. A thin, and let's say circular. What does typically a circular layer of smooth muscle do?
Contracts to squeeze?
Exactly. And like when we did that in the small intestine, or we did that in the stomach, or we did that in the esophagus, why was it making those rhythmic contractions of changing the shape? Sphincter's not a bad idea, although it's not quite that enlarged.
Peristalsis?
Yeah, it basically produces a peristalsis-like contraction to move something. What would it be moving?
Sperm?
Yeah. Why? Can't sperm move themselves? Isn't that the whole thing that's special about sperm? They're the one cell in the body that is actually self-motile.
They're still immature.
Part of it is that part of it absolutely is the immaturity. When they're in the testis, they are still immature. However, even if they were capable of a swimming motion, would you necessarily want them doing that inside the testis? Well, I'm asking the question, so what's the obvious answer?
No.
No. So but the answer then, the question then becomes why? Why wouldn't you want them swimming for themselves?
Energy expenditure?
Exactly. Basically, as we'll talk about, the way that the tail whips itself around is there is a circular motor that basically has notches in it. And each turning of that notch requires a single ATP. So basically that motor rotates around in those notches and it whips the tail around. And as it whips the tail around, it propels the spermatozoa through space. So as you could be surprised, if you wouldn't be surprised, it uses a tremendous amount of ATP to swim. And if the goal is just to move an inch forward in the testis, we don't necessarily want it using a massive expenditure of energy just to move within the ductwork system of the males. Now, it isn't until the sperm is released via ejaculation that swimming mechanism begins. Up to that point in time, it is going to be fluid pressures and peristaltic contractions of the muscles that are going to move the sperm from one place to another. So absolutely, they produce peristaltic-like contractions, they increase the fluid pressures, and these are what are going to move the sperm through and out of the testis. And we'll see what those look like in just a minute. Here we go. Like I mentioned, each tubule is about 3 feet long. So let's do some quick math, right? We'll keep it easy. 300 lobules. Each one contains 4 seminiferous tubules. Each one of those is 3 feet long. So if you were to take all of those, pull them out, stretch them out end to end, it would hurt a lot. All right. And it would also be about a ½ mile of tubing per testis. All right. As I mentioned, primary function of these seminiferous tubules, which we see in cross section here. Here we see an example of one of these, and another one here, and another one here. And if you look closely on the outer surface, again, I wouldn't use this picture for this, but since we're here talking about it right now, you can see these dark cells on the outer surface are those myoid cells. So you can see some of those myoid cells on the outer surface. Right? We can also see some blood vessels. We can also see some other cells. We'll talk about those things in a minute. But there are these myoid cells that are on the outer surface to produce that fluid pressure to help to move the sperm through the seminiferous tubule. Now, oops, now that I've done that, I have to erase this. As we know, this is also the site of hormone production here in the testis, but that hormone production does not occur in the seminiferous tubule. Instead, out here in the septum, that fibrous tunica albuginea connective tissue where the blood vessels that we see are, notice as we saw, there are these cells right here. These cells, so let me go ahead and erase the circle. These cells here in the interstitial tissue, right, in that tunica albuginea, these are the interstitial cells or the interstitial cells of Leydig, and they are the ones that are responsible for producing the androgens. Here, we get a nice illustration that shows this, and that's a good starting point. Notice we can see here on the outer surface, let's do this in purple, the thin layer of myoid cells, actually, because the nurse cells are purple, I'll use green. These myoid cells on the outer surface, that thin layer of smooth muscle cells that can produce peristaltic-like contractions, can increase the pressure. Out here, we see the interstitial cells there. Notice within the connective tissue septum, within that tunica albuginea, those are the ones that are responsible for producing the androgens. And here inside the seminiferous tubule in blue, we can see the developing sperm from basically stem cells here along the border of the seminiferous tubules to sperm-shaped cells down here near the lumen. And we'll actually identify the different stages that the cells are going through, and we'll be able to recognize them. And on the exam, if I were to point at one of these, you would actually be able to tell me specifically what cell it is. However, if you also remember when we talked about this in the endocrine system, we talked about these nurse cells. If you remember when we mentioned the nurse cells, one of the things we talked about them is that they have a kind of triangular or diamond-shaped nucleus. And so here we see the examples of some of those diamond-shaped or triangular-shaped nuclei. And all of this shaded purple that you see filling the spaces is the cytoplasm of these nurse cells. These nurse cells basically surround and support the sperm as they're developing. They also play a huge role in forming what we call the testis-blood barrier. As we know, we have defensive cells out here in our capillary that are looking for things that don't look like us to attack and destroy. And sperm cells, as they're maturing, as they're developing, do they look like us? No, they're different types of cells with different number of chromosomes and other weird things, transformations that are taking place in them. And as such, we don't want our immune system attacking and destroying them. So these nurse cells play a huge role in helping to be able to protect the testes as they develop. Notice also, basically a spermatozoa is a nuclear missile. Basically, it contains a nucleus with 23 chromosomes in it and the motor for getting it where it needs to go. So when it transforms from a cell-shaped cell to a sperm-shaped cell, there is a tremendous amount of cytoplasm that is lost during that development. And is that just garbage we want to get rid of? Is a rough endoplasmic reticulum useless?
No.
No, absolutely not. So basically what these nurse cells are, they're phagocytes that break down those components and then reuse them for the next maturing cells. If that wasn't enough, nurse cells play an important role in regulating. They release chemical signals to regulate the rate of sperm production. Didn't we talk about something else regulating sperm production? What hormone did we say was responsible for controlling sperm production? Good thing this isn't a cumulative class that we're responsible for knowing these things. All right, well, I'll give you a choice. It's either follicle stimulating hormone or luteinizing hormone. Which of the two of those was responsible for sperm production?
Follicle stimulating.
Yeah, exactly. Follicle stimulating hormone actually doesn't directly affect the sperm. It actually affects the nurse cells. And then the nurse cells control sperm production. In response, the nurse cells also produce inhibin. And what did we say inhibin did? I guess the good news is we will go over regulation of this process again, since many of you seem to have forgotten it. But if you remember, inhibin actually controls the pituitary gland and the hypothalamus to make sure that they don't release too much follicle stimulating hormone. So they play a role in not just affecting the sperm, but affecting our endocrine system as well. And if that isn't enough, we have one more issue. This interstitial cell over here all by itself has one job and one job only: to make androgens and to release it into our body. However, what's the problem we have with androgens? Do you want to just take a handful of androgens and just dump them into the blood? Why not?
Wouldn't that make everything go into like overdrive, like overproduction? And
Okay, true. Although you could argue that a teenage boy, when they enter puberty, is very much in overproduction. Remember, androgens don't cause the maturation, the development of sperm. They mature the sperm, and sperm maturation doesn't occur here in the seminiferous tubule. So it needs to be transported. The other reason androgens need to be transported is they're lipid-based. And do we want to take lipid-based stuff and just dump them into the blood and hope for the best?
No.
We want to bind them to some type of water-soluble protein so that they can be easily transported in the blood. Well, if the nurse cells weren't busy enough, just like human nurse cells have a huge job to do, and they're super busy doing it. They make androgen binding proteins. And guess what androgen binding proteins do? Not a trick question.
Bind to androgen.
Yeah, they bind the androgens and make their water-soluble proteins that bind the androgens so they can be transported in the blood. So as I mentioned, obviously the seminiferous tubule is where we make the sperm, but thanks to these nurse cells, we do a whole lot of other stuff here as well. Again, here we see, go ahead and erase my picture here. Here we see the testis in light microscopy. Again, we see the thin layer of myoid cells on the outer surface. Again, we can see that nice rectangle or elongated nucleus of the nurse cell. So there's a really good nurse cell nucleus that we can see there. And then we see the spermatozoa in different stages of development. Now, and here is under the electron microscope. To make sperm, basically we need stem cells and a unipotent stem cell. What does that mean again?
It develops into one type of cell.
Yeah, it is a stem cell that is undifferentiated, has basically one job: to divide and produce more cells. And in this case, when it divides to produce more cells, there's only one type of cell that it can produce. So these unipotent stem cells line the outer wall of our seminiferous tubules, known as spermatogonium. And like I said, their job is to divide. The testes of an average mature male produces somewhere on the order of 500 million, or 500 million if you prefer, sperm per day. Again, it's exhausting. I'm surprised we're required to do anything else. We should just be allowed to just sit and make sperm, and that should be enough. All the women should have to go to work and make the money. As I mentioned, these nurse cells are also known as Sertoli cells, or actually also succentacular cells. You may use any of those three terms as you like. And as I mentioned, they form the blood-testis barrier. They act as phagocytes to break down and reabsorb the nutrients and the chemicals. They regulate the rate of sperm development, produce the fluids that are going to maintain the sperm as it's being transported and matured, secretes inhibin, and also makes that androgen binding protein. So like I said, like real nurses, they are super busy and have lots of jobs. And notice also, again, they have these big, huge cytoplasms where they form these tight junctions with the next nurse cell, which again helps to form that barrier. We want that barrier from one to keep the developing sperm away from our immune cells. All right. Questions on that? And they're phagocytes. We did that already. Excellent. All right. Now is a good time for our first break. When we come back from the break, it's that magical time in a neuroanatomy student and neuroanatomy instructor's life where we get to talk some birds and bees about the human life cycle. And as I mentioned, as I explained to my daughter, we make the puzzle pieces that get put together. We'll have to get a little bit more sophisticated in this class than puzzle pieces, but what we are going to do is hopefully this will be review for you because you just did the Meiosis labster, but we are going to do a quick refresher of mitosis. So because we need to have to understand mitosis to understand how meiosis works. We are going to talk about these as general processes. Make sure that we're comfortable with that. And then once we are done with that, then we will see how all of these work together for sperm production. Oops, so I spell it right. So we have a busy next hour. So caffeinate, do something to be lively and interactive in the next section. All right, let's take our first break. It is 1:08, so 15 minutes makes it 1:23. We will restart. Oops. And I will start the recording at that time. All right, any questions before we take our first break? All righty, see you in 15 minutes. All righty. As I mentioned, our goal is to understand this process of gamete formation. Again, that is the generic term. And of course, when we're talking about it for males, that process is spermatogenesis. And as I hinted at right before the break, there are basically 3 steps in this process. The first is mitosis. Mitosis, of course, as we know from 430 and hopefully from the activity you just completed, is taking one cell and making 2 cells. So this is how we make more cells. Once we make more cells, we then have to convert them into special cells, the gametes. And that is the process of meiosis. And the primary process, the primary function of meiosis is twofold: to have the number of chromosomes and to mix up the genetic information so that we get variation in our offspring. At the end of this process, we went from one cell that was identical to the cell that came from it before, mitosis, to 4 unique cell-shaped cells. Of course, for male gamete formation, we then have a third step where we go from a cell-shaped cell to a sperm-shaped cell. And that is a process that takes, as you can see, about 5 weeks to occur as well. So these are the 3 big steps we need to talk about. And as I said, hopefully the mitosis and meiosis will go easy because you did the assignment. So hopefully this will make it more interactive. However, as I have said before, biology in general is like learning a different language. And if you remember way back at the beginning of 430, I said this is especially true for anatomy and physiology. And this is also especially true for genetics. And so before we get started, there's of course some vocabulary we need to make sure we understand. The first is this term, a diploid number of chromosomes. Can someone explain to me what that means in common everyday English? Apparently not.
Is that 2 complete cells?
It
Or
isn't
2
two complete
chromosomes?
cells. Okay, you've got your, I like where you're going. Di definitely means 2, right? So let me ask the question to a different way. How many chromosomes do you have in each of your somatic cells?
46.
Exactly, 46. Are all 46 of those chromosomes containing completely unique bits of information? Chromosome 1 has whether you have curly hair or straight hair. Chromosome 2 has whether you have blue eyes or brown eyes. Chromosome 3 is your blood type. Chromosome 4 is left-handed versus right-handed. Is that how it works? Do you have 46 pieces of completely unique bits of information? No.
No.
How is the information divided up on these 46 chromosomes?
By genes?
True, absolutely. But so does all 46, do they each have unique genes?
No, those are paired from each parent.
There you go. We actually have 23 pairs of chromosomes. So for instance, and I'll just draw this as a simple X. We'll talk about it in more detail in a bit, but we have those X chromosomes that we have. And for instance, if this is chromosome 1 that we got from mom, then right up here at the top may be whether you are left-handed or right-handed. And in that exact same location from chromosome 1 from dad, you have that same information, type of information, right? Is it necessarily identical?
No.
No, you could have gotten a right from mom and you could have gotten a left from dad. Of course, if you did that, would you be ambidextrous? What do you think?
Not necessarily.
No, because one is dominant and the other is recessive, right? So the right is dominant. And to be part of the proud 11%, you needed, like me, to have gotten a left from mom and a left from dad, and then you'd be left-handed. But they don't, so they can be the same, but they don't have to be. But they contain the same type of information. So what we really have is 23 pairs of unique chromosomes that contain unique information. So like I said, chromosome 1 may have the gene for handedness. Chromosome 2 may have the gene for whether or not you can curl your tongue. Chromosome 3 may contain the information on whether you've got a predisposition for heart disease, and so on and so forth. So we have 23 unique pairs, unique chromosomes in humans. Does every living organism have a total of 46 chromosomes?
Yes.
No. Some plants, or like let's say some types of sea slugs, may have a total of 20 chromosomes. And theirs would again be half from mom, half from dad. So they would have 10 pairs. There are some plants that may have as many as 96, which would be how many pairs?
48.
48. There you go. So notice if you reproduce sexually, you have paired chromosomes. And that's what diploid means. Diploid means paired. Now, notice everybody has paired chromosomes, but is the number of unique chromosomes they have always the same between us and sea slugs and plants?
No.
No. So we can't necessarily put a number here. If it was a human, we would put 23 here. 2 times 23, that would give us our 46. If it was a plant, we would put 48 in here, and that would be that. If it was a sea slug, we'd put 10 here. But rather than have to put every number of every living species that could be, what could we put here instead? How about a placeholder, right? Like an X or like an N, exactly. N just stands for the unique number of chromosomes. Exactly. So when we talk about the diploid number of chromosomes, it's 2n. And for humans, 2n equals 46. So of course that tells us we have 23 unique chromosomes. Again, you already knew you had 46 chromosomes. You already knew you had 23 pairs. There's no new information here. We're just adding some vocabulary to it. This value of 2n. The reason this is important is because, as I mentioned, when my wife and I put our puzzle pieces together, did I want to provide 46 chromosomes and she provided 46 chromosomes and we mashed those together to make a baby?
No.
No, because then she would have 2 heads and superpowers and all sorts of other bad things. So what we need to do is we need to have those. Part of meiosis is splitting those in half till you have a haploid number of chromosomes. Of course, for humans, that haploid number would just be 1 copy of each unique chromosome. So of course, for humans, that would be how many? 23. For the sea slug, how many would that be?
10.
And for the plant, it would be 48. And so again, we're going to represent that with just an N. So diploid, 2N, 2 copies of each unique chromosome. Haploid, 1N, 1 copy of each unique chromosome. And so our gametes need to be haploid. I give my 23, my wife gives her 23, and we have a baby that gets 46. And shares our genetic information. Has my earlobes and my wife's temperament, right? And all the other fun things that go in between. So the only haploid cells are the sperm and the egg. Now, notice, as I mentioned, in my somatic cells, I have 2 copies of chromosome 1 with a specific piece of information. These 2 these two chromosomes, one that we get from mom and one we get from dad with the same type of genetic information, are what we call homologous chromosomes. So my chromosome 1 from mom, my chromosome 1 from dad, those are what we call homologous chromosomes. I have one copy of chromosome 2 from mom, one copy of chromosome 2 from dad. They're homologous chromosomes, or what we call homologs. But if you remember way back in 430, when we talked about mitosis, you will remember that when it is time during, let's say it this way, during the S phase of interphase, our DNA is replicated. So we have two copies. Those two copies are sister chromatids. If you remember some vocabulary, here's one copy of the genetic material and the other copy of the genetic material. These two identical pieces of genetic material are the sister chromatids. They're held together by a special protein, which is where we get that X or H shape. Does anyone remember the special protein that holds the sister chromatids together? Excellent. Centromere. And remember, during mitosis, there were special proteins on the outside of the sister chromatids that are microtubules or spindle fibers could connect to move them around through space. And does anybody remember what those special proteins to move the chromosomes around inside of the cell were called?
Kinetochores.
Yeah, there we go. Excellent. Kinetochores. Oops. There we go. Kinetochores. Perfect. So one chromosome is made up of these 5 things. 2 sister chromatids held together by a centromere, and each sister chromatid has a kinetochore. So these 5 things together basically is one replicated chromosome. We call it one chromosome, but that makes things tricky because when we're in the cell and we have that one copy by itself, that one copy by itself is called a chromosome. So this right here, again, it's obviously loose, but this right here would be one chromosome. But this over here is one chromosome as well. So we need a way to tell those apart. We need a way to indicate that this chromosome is replicated. And the way we're going to do that, the nomenclature we're going to use is NR. That R means that it is replicated. So the difference between 2N and 2NR is that 2N is just a single chromosome, 46 of them. 2NR would be 46 replicated chromosomes. This and homologous are really the only new pieces of information we're adding to this. So hopefully that's not too much and that makes some sense. Any questions on that? All right, excellent. Actually, hold on, I want to save this because it's going to go away. Oops. No, I meant to push. All righty. So briefly and quickly, mitosis. Tell me what the goal of mitosis is.
To form a new cell.
Excellent. So basically, we want to divide one cell to produce two cells. But what can you tell me about those two cells?
Identical to the first cell.
They're going to be identical to each other and to the original cell. So in the case of our human cells, we have one cell with 46 chromosomes inside of it. We want to divide it into two cells. Both of which also have 46 chromosomes in them. And how many steps are there in this process?
4.
Excellent. What are they?
Prophase, metaphase, anaphase, and telophase.
Excellent. Yep, exactly. Errol, your PMAT, which you have in your bathroom. Exactly. Now, technically, those would just give you two nuclei. So what else do we need as well? True. We'll talk about prometaphase in a second. But well, again, if we go through prophase, metaphase, anaphase, and telophase, we would end up with one cell with two nuclei. We want two cells, so we need some other step as well.
Cytokinesis.
There we go. Cytokinesis is where we split the cytoplasm, dividing the one cell into two. Now, you are correct, Ervek, that if you remember back in the textbook with prophase, a lot of stuff happens. So at first they talked about prophase, and then they talked about early prophase and late prophase. And then after that, it got to a time they decided, hey, we need to sell new textbooks. So rather than just talking about early prophase and late prophase, let's make early prophase, and late prophase we'll name some new phase that is between prophase and metaphase. And because they're incredibly original, they named it prometaphase. Exactly. Again, all of those are subtlenesses to the process. Remember, as we talked about, you could say early and late prophase, or you could say prophase and prometaphase. We were fine with it either way. Since we're doing this a little bit more simply, we'll just go with prophase and metaphase for now. So you're right. If we were talking about mitosis more in detail, we would want to make that, that notation of either early or late prophase, or throw that prometaphase in there as well. Excellent. So that is, in a nutshell, what's going on. And of course, as we know in prophase is when we, there we go, form the chromosomes. See if I can sneak this in here. This is where we line up. And I'll just put the chromosomes, just that. Anaphase is where we split the chromosomes. Telophase is where we unwind the chromosomes. And then, as we said, cytokinesis is where we split the cytoplasm. So in a nutshell, that is mitosis. hopefully, again, because you guys did the assignment, we don't have to go too much more in depth into that. So questions on mitosis and understanding mitosis, because if you don't understand mitosis, the rest of this is going to hurt. So let's make sure we understand this before we move forward. Questions on mitosis? All right. If we are comfortable with mitosis, then what we can do next is we can talk about meiosis. What is the goal of meiosis?
To create 4 haploid daughter cells.
Exactly. We want to divide one cell into 4 haploid cells. And do we want these cells to be identical to the original?
No.
No, if they're haploid, they can't be. So we want 4 haploid cells that are unique to each other and the original. To do this requires 2 divisions. Now, the good news is at its core, the process is similar to mitosis. So we can easily divide this into 4 stages. Oops. No. And they're even named the same. Prophase 1, metaphase 1, anaphase 1, and telophase. And guess what the next 4 stages are called?
Phase 2, metaphase 2, anaphase 2, and so on.
There you go. See how easy that is? Perfect. All righty. Now, I don't think I left myself enough room for all of this. So the first division is meiosis I. And it has those stages: prophase 1, metaphase 1, anaphase 1, telophase 1. This, as I mentioned, obviously by the names, are going to be similar to mitosis. However, the goal here in meiosis I is to split the chromosomes. We want to form 2 haploid cells. So that is our major goal in here. Prophase is going to start in a similar fashion. If you think in terms of what is happening during mitosis, if you remember, in prophase, basically 3 big events occur. The first is that we break down the nuclear envelope. I think I'm going to have to write this smaller. We break down the nuclear envelope. We fold up the chromatin into chromosomes. And the third thing that happens is the centrioles migrate to the poles of the cell. So if we think in terms of what's going on in our cell, here we've got our cell in here. Our nuclear envelope breaks up. Our spindle, which we'll draw in green here. Actually, no, I'm gonna use black. Our centrioles reach the poles and they start producing their spindle fibers. And our loose chromatin folds up into tightly packed chromosomes. Now, if this was a human cell, I would have to draw 46 different chromosomes in here. And clearly, I'm not going to fit that in here. So what I'm going to do instead is draw 4. Here's chromosome 1. Here's chromosome 2. Here's chromosome 3. And here's chromosome 4. So these are all the things we would expect to see happening in a normal prophase. So far, so good? All right, but here is where prophase 1 of meiosis is different from mitosis. Notice, and I did this on purpose, I used 2 reds and 2 blues, because the other big thing that happens here during meiosis 1, prophase 1, is our homologous chromosomes find each other and they attach. A special protein called synapsin basically comes in here and zips them together. So this protein zips them together, binding the homologous chromosomes to each other. When this occurs, they form structures that we call tetrads. If you think of mitosis, every chromosome is for itself. It's doing its own thing. It's independent. But here, our homologous chromosomes are going to find each other. And so notice, whereas in prophase we formed chromosomes, in mitosis, here we're forming tetrads. What did we do in metaphase? In mitosis?
A line in the middle of the cell.
Yeah, we lined up chromosomes. Well, here in metaphase 1, we line up tetrads. So here, our tetrads are spindles attached to the kinetochores and move the tetrads to the metaphase plate, to the equator of the cell. Randomly. There's my cell. Here's my spindles. My centrioles. Now, when my tetrad lines up is, let's say the dark colors are mom's. Is mom always going to be on top and dad going to be underneath?
No,
No. How these line up is completely random. One time, mom could be on top, dad could be on the bottom. The next time, dad could be on top, mom could be at the bottom. And how the red ones line up, is that going to have any effect on how the blue ones line up?
no,
Yeah, if I have 2 quarters and I flip the first quarter and it comes up heads, does that have any effect on what the chances of getting a head or a tail on the next one is going to be?
nope.
No. So this is completely random, what we actually call random assortment. Now, how many different combinations can that give us? If I have 2 quarters and I flip them both and they both are independent, how many different possibilities can I get from them?
Four.
Yeah, 2 heads, 2 tails, head, tail, tail, head. Right? 4 different combinations. Now, I was able to figure it out on my fingers, but there's another way you can figure it out as well. How many options are there with heads and tails?
2.
2. How many different coins did I throw?
2.
So 2 to the 2 power equals 4. Well, with our chromosomes, mom can be on top or dad can be on top. We still have 2 possibilities, but how many tetrads are there going to be inside of our cells?
23.
There you go. So all of you have these smart devices in your pocket or sitting on the counter next to you. Someone tell me what 2 to the 23rd power is.
$8 million, 388,608.
Exactly. Over 8 million different combinations. So every time these chromosomes line up, there is one of 8 million different combinations that they can form. So do you and your older or younger sibling look identical to each other?
No,
No, exactly. Because of this uniqueness, the unique way that these mix and mangle up gives us over 8 million possibilities. So you look similar to your siblings, unless you have an identical twin, obviously that's different. But your older or younger siblings, if you have a fraternal twin, you look similar, but you're not identical. And that whole Patty Duke thing, total BS. Because of this random assortment of how these things mix up. This gives us a huge genetic variation. Which is vital for the propagation of our species. All right. Excellent. Questions on that? All right. Prophase, we formed the tetrads. Metaphase, we lined up the tetrads. So guess what happens in anaphase?
They start to move towards opposite poles.
Exactly. Notice everything we did to chromosomes in mitosis. In meiosis I, we're doing to tetrads. So remember that synapsis protein that was holding the tetrads together inactivates, and whoops, the replicated chromosomes separate. Then the spindles those tetrads get pulled, pardon me, those replicated chromosomes get pulled to the opposite poles. Again, we get that nice distinct V shape, just like we saw in anaphase. As our replicated chromosomes are pulled towards the opposite poles. Of course, remember, this is also when cytokinesis begins. Starting that cleavage furrow as that actin filament, that actin ring is pulling on the cell, starting to pull it together, dividing the cell in half. And so then in telophase and after cytokinesis, we have two cells. And even though, here, let's do it this way. Even though I've been dividing it top and bottom, I'm going to cheat and put them side by side just to give myself a little more room. I have two cells. These cells are both going to reform their nuclear envelopes. Our loose chromatin unfolds in, our tightly packed chromosomes unfold into loose chromatin. But again, I'm going to just put them in here, bound together so that we can see them more clearly. Wow, that was bad. Excellent. So at the end, we have two cells. And what could you tell me about these two cells?
They have half of the amount of chromosomes.
Exactly. Notice these cells are, we have two cells that are haploid. Each has only one copy of each unique chromosome. So both have one copy of chromosome 1, both have one copy of chromosome 2, and the chromosomes are still replicated. So notice we went from a cell that started as 2NR, right? We had a 2NR cell. How do you think you would identify these cells?
1 NR?
Yeah, 1NR. And when you're doing 1NR, do you have to put the 1?
No.
No. So you could also just say NR. The 1 is a great placeholder if it helps you to remember. So that is totally acceptable. But anytime, you don't have to say 1X in algebra, you can just say X. So we could do the same thing here. We could just do NR. So one NR or NR, both of those would be acceptable ways of identifying those cells. And so here, notice our goal was to split the chromosomes to form our 2 haploid cells, and we have done that. And we did that by using tetrads. All right. We formed tetrads, we lined up tetrads, we split the tetrads. And we got that genetic variation thrown in as well. All right. Questions on that?
So in Meiosis I, the chromosomes don't replicate.
Well, remember, replication occurs during S phase of interphase. So that happens before the division starts. So coming in, they're already replicated. All right, this cell here was replicated to give us 2 cells that have 46 chromosomes. All right, that's how you go from 1 cell with 46 chromosomes to 2 cells with 46 chromosomes. You have to replicate the DNA first so that each cell gets a copy of it. So that's why I like that R as a placeholder, because this tells me this one's replicated. It has 2 copies, so we can give 1 copy to each.
But the cells end up haploid.
Yes, because we want them to be haploid. We started, so again, if these were human cells, each of these would have 23 chromosomes in them. We started with 46 replicated chromosomes, and we get 23 replicated. Think of it this way. I always like to think of this in terms of a deck of cards. How many cards are there in a deck of cards?
52.
52. Excellent. If I have one deck of 52 cards and I have 2 daughters, and I want to give them each a deck of cards, can I divide my one deck into 2 functional decks of cards that are both going to have ace of spades and both going to have queen of hearts and both going to have a 3 of clubs?
No.
No. What I need to do is I need to make a copy of that deck of cards first. So essentially I have 2 decks of cards. However, when I have those 2 decks of cards, do I just shuffle them together and then give 52 to one and 52 to the other? Would that be a good way to do that?
No.
No. What I need to do, if they want to have functional cards, I need to make sure that both of them get an ace of spades and both of them get a queen of hearts. So what I do is I put the 2 ace of spades together, and I put the 2 queen of hearts together, and I put all the pairs together, all the identical cards together, so that I can give 1 to one and 1 to the other. And that way, each of them gets a functional deck, 1 copy of the card that they need. And so that's basically what mitosis is. All right. Here, I'm putting them together. So the 2 aces go to, of spades go to one, and the 2 queens go to the other. Right here, I'm replicating them. So notice I've got half the deck of cards, because I'm taking my half a deck of cards and my wife's half a deck of cards and putting them together to make a full deck. So here I've got 2 replicated cells, because am I done? Have I met my goal of meiosis?
No.
No. So luckily, there is a second division. Our first division, Meiosis I, our goal was to make the haploid cells. And the good news is Meiosis II is essentially identical to mitosis. Where we want to split replicated chromosomes into 2 cells. So in prophase 1, again, we form our chromosomes. In both of our cells, and now I'm going to put them top and bottom again to have enough space. Actually, I lied. Maybe I'll put them side by side again. So again, we're going to break down the nuclear envelope. We're going to fold the chromatin into chromosomes. And we're going to migrate the centrosomes to the poles. Again, our nuclear envelope breaks down. Our chromatin lines up. So here we have our chromosomes form independently. And again, our spindles attached to the kinetochores. Excellent. What happens in metaphase? Come on.
They line up.
Exactly. Each chromosome lines up independently. On the metaphase plate. Our spindles move them, enlarging or breaking down to move them to the center, to the metaphase plate, to the equator. So this is where we line up the chromosomes. So what do we do in anaphase?
Sister chromatids separate and beginning of cytokinesis.
Exactly. We're going to split the chromosomes. You are 100% correct. Our kinetochore, pardon me, centromere inactivates, and our kinetochores draw the chromosomes to the opposite. And you're also correct, cytokinesis is going to begin. Oops. Cleavage furrow forms as our actin ring pulls and squeezes the cell. And so what is the end result of all of this?
Four haploid cells.
Exactly. Good. Nuclear envelope reforms. Our chromosomes unwind. Cytokinesis ends, and we have 4 unique haploid cells. And notice each of these we would just call 1N, or as we mentioned, we could just use the term N. So 2 divisions, 4 cells, haploid, half the number of chromosomes, and all completely unique. Questions on that? You know how much I enjoy and appreciate my stunned silences, but I'll ask the question again. Does anybody have any questions or any problems with what we've just done and said? Well, I'll say it again. We have 4 unique chromosomes, 4 unique cells. Do we really have 4 unique cells? Are those cells, they're definitely haploid, they definitely have half the number of chromosomes, but are they truly unique?
No.
2 of them are unique, but 2 of them are identical to each other, right? So we really haven't reached our goal. So questions on what we've done here before we add one more step. All right, stunned silences, of course, mean we understand this material perfectly. Dr. Slutsky, go faster. So let's do that. Excellent. Way back here in prophase 1, one other thing happened as well. There you go, Aravec, you hit it on the head. A very special process known as crossover. Basically, what happens with crossover is that as the tetrads form, pieces of the chromosomes switch positions. So let's look at just one example of this. Let's say, for instance, down here, the leg of this first chromosome. Actually, we'll use the red up here. This portion of, let's put a box around it. These two changed positions. What that would mean is over here, a little bit of the pink chromosome was now over here, and let's make this bigger so it stands out. This is now a little bit pink over here. And this has got a little bit of red right there. So when they separated, this has got a little bit of red on it. This has got a little bit of pink on it. So that this has a little bit of pink on it. And this has a little bit of red on it. And the same thing here, little red here, little red here, little red here. So this one here is half red and half pink. The same thing for this one. This has a little bit of pink down here, 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 a little bit of pink down here. And this one has a little bit of pink, half pink and half red. Now, with just that one switching of that one little piece of genetic material, now do we have 4 unique cells?
Yes.
Yeah, absolutely. And notice that was just with one crossover occurring. Dozens of crossovers can occur during this phase. And this is how we mix and match our information, right? Your daughter, your son don't have all of the characteristics of your grandmother and all the characteristics of your mom's grandfather or something along those lines. They're a jumbled mix of characteristics. And so it isn't just how they line up that causes those unique characteristics, but it's also this constant crossover that takes place during prophase 1 that gives these unique cells. Now, I've done it here on the whiteboard, but let's take a look at this with the pretty pictures from your textbook. Again, here we're emphasizing the differences between mitosis and meiosis. Notice this one, like mine, is a simplified cell that has 6 total chromosomes. So this is a cell, this cell right here is 2N. And in this case, the N, the unique number of chromosomes would be 3. So basically this is mom 1, sitting next to dad 1, sitting next to mom 2, sitting next to dad 2, sitting next to mom 3, sitting next to dad 3. 3 unique chromosomes, 2 copies of it. So this is a diploid cell. 2N. Notice the first thing we have to do is replicate it. Technically, how many chromosomes do we have right here?
12.
No, technically this is 6. When they're held together by that centromere, this is just considered 1 chromosome. So this is considered 1 chromosome, but this is also considered 1 chromosome. As long as they're held together, they're considered 1 chromosome. That's why I emphasize that 2 and R. That R lets us know that it has been replicated, that it's 1 chromosome, but it's 1 chromosome with 2 copies of the genetic information. And we have to have it replicated so that 1 copy can go to each cell. 1 copy of the black goes to each cell, 1 copy of the gray goes to each cell, and so on and so forth. So now we have 2 cells with a haploid number of chromosomes. That makes sense? All right, let's do the same thing for meiosis. Starting with meiosis, we again start with a cell that is diploid, and it needs to replicate. So all of our chromosomes are now replicated, but notice the paired copies, mom 1 and dad's 1, mom's 2 and dad's 2, mom's 3 and dad's 3, come together to form that magical structure known as a tetrad. Notice we still technically have 6 chromosomes because they're held together, but we also have 3 tetrads. And our goal in meiosis 1, remember, is to split the tetrads. And so that's exactly what we do. 1 of the chromosomes from the first tetrad goes one way. 1 of the replicated chromosomes from that tetrad go the other. And the same thing happens for all 3 tetrads. And now that we split the tetrads, we have cells that are haploid, but their chromosomes are still replicated. Which is why we refer to them as NR. They are haploid, but they are replicated. The advantage of them being replicated is they can then still divide. 1 copy of chromosome 1 goes to 2 cells, 1 copy of chromosome 2 goes to 2 cells, and so on and so forth, giving us a total of 4 haploid cells with just 1 copy of the genetic material. So these are N. So that's our goal. Remember, our goal of meiosis 1 is to split the tetrads, and the goal of meiosis 2 then is to split the replicated chromosomes. Which, if you think about it, is exactly what the goal of mitosis. Mitosis's goal is also to split the replicated chromosomes, which is why I said mitosis 2 is basically identical to meiosis. Pardon me, meiosis 2 is identical to mitosis. Now, obviously, we need to know this in more detail than this, so let's actually look at the process of meiosis. Meiosis starts in prophase 1, as we see here. Again, remember there are 4 main goals in prophase 1. Goal 1, migrate the centrosomes to the cores, to the poles. Goal 2, break down the nuclear envelope. Goal 3 is to condense the chromatin into chromosomes, but goal 4 is to form the tetrads. And while we're forming those tetrads, remember we can increase our genetic variability by having that crossover occur. So prophase 1, we form the tetrads, which leads to metaphase 1, where we line up the tetrads. Notice our crossover here, we've gotten 4 crossovers that have occurred on this particular one. Crossover 1, crossover 2, crossover 3, crossover 4. Anaphase, the synapsis inactivates and our replicated chromosomes separate. Our tetrads become replicated homologous chromosomes, going to opposite cells. And after cytokinesis, we have 2 haploid cells with replicated DNA. And because the DNA is replicated, we need meiosis II. Prophase, migrate the centrosomes, break down the nuclear envelope, form the chromosomes. Metaphase 2, line up the chromosomes. Anaphase 2, separate the chromosomes until telophase, where you have, and again, obviously this happening in both cells, they just showed it in one, gives us a total of 4 cells. With 1 copy of the DNA. And because I like our visuals, here's another great one. This is the one actually from your textbook. Your textbook does an excellent job. So if the way I've been explaining it to you, I sound like the teacher on Peanuts, then I strongly encourage you to read it in your textbook. Your textbook does an awesome example. Notice your textbook, like me, just did 4 chromosomes, because putting 46 in here would be a bear. Like me, they just did 1 crossover. But again, prophase 1 of meiosis I, break down the nuclear envelope, migrate the centrosomes, form the chromosomes, then form the tetrads. And while the tetrads are forming, we can have crossover. Prophase 1 is when we form the tetrads. Metaphase 1 is where we line up the tetrads independently, flipping a coin. Notice here, yellows are both on the right, on the left, blues are both on the right, but it could just as easily go the other way. This could be flipped, that could be flipped. It's totally random. Tetrads line up in metaphase 1, tetrads separate in anaphase 1. So 1 copy of the replicated chromosome goes one way, 1 copy of the replicated chromosome goes the other. Then after telophase, we reform the nuclear envelopes. Cytokinesis divides the cell in half. We have 2 haploid cells with replicated DNA, which we need to divide with meiosis II. And meiosis II, prophase, metaphase, anaphase, and telophase 2, is basically just like mitosis. Chromosomes form, nuclear envelope breaks down, chromosomes migrate. Each individual chromosome lines up. Each individual chromosome is separated. each individual group of chromosomes get their own nuclear envelope and get divided into their own cell. And we have 4 haploid cells. And again, notice with just one crossover, these cells are unique from the original and also unique from each other. 4 unique cells with half the number of chromosomes to produce our gametes. And again, it is important to understand that this is true for both males and females. This is the basic process for both males and females. Meiosis gives us 4 haploid cells that are unique from each other and unique from the original. All right, questions on that?
Yeah, so tetrads are like double chromosomes.
It isn't like we're doubling the genetic material. It is like finding the pairs and putting the pairs together. So you have 1 copy of chromosome 1 from mom and 1 copy of chromosome 1 from dad. Then during S phase, they get replicated. Then during meiosis I, they find each other. And so basically it is, remember, these are homologous chromosomes. If you go back, I erased it already. If you remember when we were talking, we have chromosome 1 from mom and chromosome 1 from dad, and both of them have the same genetic material. They have up here at the top, they have your handedness, whether you're going to be right-handed or left-handed. So these are your 2 genes that determined whether or not you were left-handed or right-handed. All right. Let's say this was my dad, for instance. My dad is right-handed. So one of these genes is a big fat R, and one of these genes is a big fat L. When he was making his gametes, these need to separate into different cells. We want to get them into different cells. So he can give one of these to me. And in this case, for me, which of the ones did I get? Did I get his mom's or did I get his dad's?
His dad's.
Yeah, because I'm left-handed. All right. My sister is right-handed. So guess which one he gave her?
Your mom's.
His mom's, right? These are his chromosomes. My mom is easier. My mom is also left-handed. So for my mom, we'll put my mom over here. She has 2 copies, one that she got from her mom, one that she got from her dad. However, my mom is left-handed. So did it really matter which one she gave me?
Nope.
No, it doesn't matter which one she gave me. She gave me an L. Dad gave me an L. I have 2 L's. I'm left-handed. Right? She may have given her mom's to my sister, but my dad gave his mom's as well. She got an R and an L. She's left-handed. So the whole point of putting the tetrads together is so you can keep track of the same information, those homologous chromosomes, so that everybody gets one version of it. That make sense? Hopefully. Again, your Master A&P's got some great videos. Your book does a great job of describing this. So again, if you're having trouble with this, then definitely look at those other resources. Because like I said, I do the best that I can to describe these things, but sometimes hearing it in a different voice, having someone else describe it, or watching the animations and stuff like that can help to make sense of it. So if you're struggling with this from me, then I encourage you to look at that, because this is definitely something, describing meiosis is absolutely something that could be an essay question on the exam. Now, as I mentioned, both males and females use meiosis for the formation of the gametes. However, is the formation of the gametes in males and females identical?
Nope.
No. So Now that we understand the basic way of doing this, we now or the general, let's go back and think of it that way. This is the general way for both males and females. Now, what we're going to do is talk about the process specifically in males. And remember, as we mentioned, that process specifically in males is known as spermatogenesis. So this is what we will talk about next. However, what I will do is I will go back to my illustration of this. Again, I don't know if this is necessarily the best one, but at least it's all in one place for you to look at it and think about this. And we will take our next break. It's 2:42 right now. So we'll come back at 2:57. And we'll start the recording at that time. All right. So we'll give this some time to sink in, but any questions before we take our next break? All right, see you in 15 minutes. All righty. Any questions before we get started?
Professor, can I ask a question about the lap stimulation? Really quick?
Sure.
So while doing that lab, I've noticed something interesting. There were two clips, in short clips in the meiosis. The first one was showing, it was about the IVF, and the first one was showing how they fertilize the egg.
Mhm.
The process, and the second one was showing how they load already fertilized egg into the catheter so it can be placed into the uterus.
Yes.
So the interesting or maybe weird part was that they didn't actually took the entire embryo into the catheter. They took one cell, a little piece of the embryo into the catheter. So does that really how it works?
No. So I did it during the break, so I don't remember it that closely, but they do. So what I think maybe typically they will mature multiple eggs at the same time, and then one or a couple of those, the ones that they think are best, will be the ones that they will then insert into the uterus. So they get it to that blastula stage, which we'll talk about when we get to development at the end of this, and insert it as that. They wouldn't insert just one cell, they would insert the ball of cells that have been formed. So they may form multiple, they may mature multiple eggs at the same time to hope to get to a point, but typically just the two or three that they think are the most, or that look the healthiest or the largest or the most developed will be the ones that they will insert. The rest they can actually freeze and then potentially insert later, but they wouldn't just do a part of the blastula.
Well, this one was like close up, and they injected just only one sperm into one egg, and then they showed after a couple days it was divided into maybe like 8 cells, and they like pull just one cell out of that embryo.
Well, at that point it would be a morula, or those are the blastocells, the individual cells that are dividing. My guess is that what they're doing is an oversimplification. You've got the right idea. They are going to put the sperm and the egg together, but they don't just put one sperm with one egg together. That wouldn't be as practical. So I think they were just showing that for simplicity reasons. But yes, once you have that first base ball, that zygote divides into 2 cells, they divide into 4, they divide into 8. If you were to separate those, they still are omnipotent enough where they can develop into full individuals. In fact, that's how identical twins are formed, right? That ball of cells separates very early in the process and they develop. So theoretically, you could take out a single one of those blastomeres from that developing zygote that is dividing and you could get a person out of it. But in all practicality, you wouldn't do it that way because it would have a low likelihood of success.
Okay. Yeah, the weird part, because I thought maybe, is it possible to get multiple babies from one zygote? If that's the case,
Yeah, that's exactly, again, when you have identical twins or identical triplets, that's exactly what happens. That one egg and one sperm form a ball of cells, and those balls of cells separate and become individual people. So that's exactly how you get identical siblings.
Okay, thank you.
So yeah, so that does happen in real life. All righty, great question. Any others? And we will, like I said, at the end, we will talk about development. So we will come back to that, and maybe I can answer some more questions about that when we look at and talk about that process. All right. Our goal then now is to talk about the specific case of spermatogenesis. Spermatogenesis is going to be the process by which we produce our sperm. And there are 3 steps in this process. And those 3 steps are first mitosis, second meiosis, and then third spermiogenesis. The goal of each of these is such. Our goal of mitosis, as we know, is to produce many cells. Right? Or more specifically, to divide one cell into 2 cells. So if a male is going to produce 500 million sperm during the course of the day, then obviously we need lots of cells to be able to do that. Meiosis is the process where we are going to be able to produce those 4 haploid cell-shaped cells. And then spermiogenesis will be the process where we convert the cell-shaped cells into sperm-shaped cells. Oops. Excellent. And remember, this all occurs in the seminiferous tubule. So let's start with the wall. Here is the border of our seminiferous tubule. And of course, as we know, on the outside, there's going to be those myoid cells that we talked about. And then hanging out here, away from it, might be that interstitial cell that is going to produce the androgens. Now, this process, when does it begin in males?
Puberty?
Yeah, absolutely. When does it end?
Never.
Well,
After death?
about death, yeah, about 15 minutes after they're dead, right? Absolutely. So yes, males, once this process begins, it is a continuous process basically for the rest of their life. May there be minor declines in the amount of spermatozoa that are produced? Yes. Does there have to be? No. And we see plenty of instances, especially in celebrities, of males that are far, far too old to be having children and taking care of children, having children. So we see plenty of examples of that because this process is basically continuous and stays at a high level pretty much till death. As we talked about, it is going to start with a stem cell. And as I mentioned, I guess we're gonna have to make this smaller now. As we mentioned, this unipotent stem cell always hangs out here. next to the border of the seminiferous tubule, and it is our spermatogonium. And this unipotent stem cell, the spermatogonium, undergoes mitosis. How many divisions in mitosis again?
One.
One, excellent. I'll put this over here. And how many cells does it produce as a result of this?
2.
And what do we know about these two cells?
They're identical to the parent cell.
Exactly. So basically we go from a cell that is 2N to two cells that are 2N. Identical to each other and identical to the original. And now that I'm looking at this, I may have made these all too big. So let's make them a little smaller. So we get two identical daughter cells. And these two identical daughter cells are basically called the A cell and the B cell. While these two cells are identical to the original and identical to each other, they ultimately have two completely different goals. The A cell basically stays at the border of the seminiferous tubule and basically becomes a spermatogonium. It essentially replaces the cell that made it. So we started with one stem cell, it divides into two cells, and we have one stem cell remaining. In this fashion, this process can continue forever, because the stem cell, when it divides, is always replacing itself. However, the B cell migrates away from the border of the seminiferous tubule and it matures. And part of that maturation is undergoing interphase and replicating its genetic material. And so this B cell basically grows big, grows large, replicates its genetic material, and becomes what we call a primary spermatocyte. So we have one primary spermatocyte. So one spermatogonium produces one spermatogonium and one primary spermatocyte. This primary spermatocyte then undergoes meiosis I. How many divisions do we get with meiosis I?
One.
One division. And how many cells do we get?
Two.
Two cells. Excellent. And what do we know about these two cells?
They're haploid.
Oops, that didn't need to all be in capitals. With replicated DNA. So our two NR cells have now become, oops, NR. And these two cells we call our secondary spermatocytes. Now, why are they called secondary spermatocytes?
Because they come after the primary.
Yeah, primary came first, secondary came second. Absolutely. And it's the same thing here, primary, secondary. However, the convenient thing is we have one primary cell, primary is first. We have two secondary cells. Secondary is second. So that makes it convenient. So we have these two secondary spermatocytes. These do not replicate the DNA. It's already replicated. So they just go straight into meiosis II. Each of these cells divide how many times?
Once,
Yep. And how many cells does each one produce?
2.
So as a result of that, we get a total of 4 cell-shaped cells, all with half of the genetic material. Whoops. And these 4 haploid cell-shaped cells we call our early spermatids. So we have 4 early spermatids. Lastly, these 4 early spermatids undergo spermiogenesis. And after undergoing spermiogenesis, we get 4 sperm-shaped cells. With a head and a tail. And we'll talk about the anatomy of this in more detail in a minute. Mitosis, meiosis, spermiogenesis. We go from 1 stem cell to 4 sperm-shaped cells. Start at puberty, end at death, and this whole process takes approximately 70 days. From stem cell to sperm-shaped cells. Let's do this again. Again, notice there are 4 steps in this process, or 4 different cells, I should say, stages in this process: the spermatogonium, primary spermatocyte, secondary spermatocytes, and then our spermatids, both early and late spermatids. So I guess we should say that I didn't do that on the S one. Once they're sperm-shaped cells, these are now late spermatids. It wouldn't be completely wrong to call these spermatozoa yet, but the key, what I like about staying with the term late spermatid is that these are immature spermatozoa. They are shaped like sperm cells, but they can't function like the sperm cells yet. They're not capable. If you were to pluck one of these out and rub it on an egg, it wouldn't be able to do anything. It would not be capable of fertilizing that egg. So there's nothing wrong with calling it a sperm. Whoops, spelled that wrong. spermatozoa, but I like late spermatids because it reminds me that there's still work to do. All right, excellent. Let's go back to the pretty pictures. And then ultimately the mature sperm, which occurs actually outside of the testis. Here are the pretty pictures. And notice again, your book doesn't put the R here, well, here actually, for the reminder that it is replicated DNA. But again, we have that spermatozoa production begins at puberty, ends 15 minutes after they're dead. Our spermatogonium, our stem cell, undergoes mitosis, producing two identical daughter cells. The A cell, which stays at the basal surface of our seminiferous tubule, and the type B cell, which migrates away. So when a cell divides, it is always replacing itself, allowing this to be a continuous process, allowing a male to produce the gajillion sperm he produces during the course of his life. But the second type B cell migrates away from it and matures, replicates its DNA. And if you think about it, this cell basically has to become 4 cells. So the other thing that happens to this cell is by far the largest of all of the cells. Remember, as I mentioned, we're going to be needing to recognize these and identify these histologically. These are probably the easiest to identify because they're not right at the border of the seminiferous tubule, and they're by far the largest of all of the cells in the seminiferous tubule. This primary spermatocyte undergoes meiosis I, causing 1 division, giving us 2 haploid cells that are again replicated. And those 2 replicated haploid cells are our secondary spermatocytes. Our secondary spermatocytes undergo meiosis II, forming 4 haploid early spermatids. So now we've reached our goal of having 4 haploid cells that are unique from the original and unique from each other. But as we mentioned, we need to convert them into chromosome missile. And to do that requires a process called spermiogenesis. Now, your book actually does an excellent job of describing the process of spermiogenesis. It's a process that takes about 24 days, where, as we mentioned, we shed the most of the cytoplasm and basically have this chromosome missile as a result of it. If you're interested in understanding that process, then feel free to read about it, but I'm not going to make you describe the process of spermiogenesis in our for the exam. So you just need to know the name of it and what occurs, and you need to know the end result. The end result is our spermatozoon. And as you can see from this illustration, and we'll cheat and we'll go to the whiteboard for this. Spermatozoon basically has 3 different regions to it. The first is what is known as the head, or the nuclear region. This is the enlarged portion. This is what contains the nucleus. Or let's be more specific, the haploid nucleus. Or if we want to be more fancy, the 23 chromosomes. But there is another specialization to the nuclear region as well. At the distal end of the head is this enlarged structure known as the acrosomial cap. For those of you who have taken me for 430, and I've probably done it here in 431 as well, often when describing the anatomy of something, when we talk about the outer layer, I jokingly refer to it having a candy-coated shell. All right, that's a common term that I use a lot. Well, guess what? The egg actually literally has a candy-coated shell on its outer surface. There is a glycogen-rich protective layer on the outside of the egg. I know every video, every movie, every joke they've ever talked about, it's always the first sperm that gets to the egg gets to fertilize it, right? Nothing could be further from the truth. There is nothing more American than the fertilization of an egg. Because what happens is the first 200 sperm get there, kill themselves, beating away at that barrier on the outside. And that lazy sperm who comes lollygagging in 3 hours later, after they've done all the work, he's the one that wiggles through the hole that they made, fertilizes the egg, and gets to win at the end. The majority of the sperm, many sperm are required to fertilize the egg, and primarily to defeat those defenses. And the way they do that is with this acrosomial cap. It's basically an enlarged structure that contains a bunch of hydrolytic enzymes so that they're able to digest that candy-coated shell. So lazy boy can come and fertilize the egg at the end of the process. America. All right. The second portion of the sperm, I don't like purple anymore, is the midpiece, or what is also known as the motor region. As the name would indicate, this is the area where we produce the propulsion. As I mentioned, there's a rotary motor. I don't know, that looks right. Rotary motor that requires massive amounts of ATP. Well, luckily for us, during this process, massive numbers of mitochondria have wrapped themselves around this motor piece. So it is filled with coiled mitochondria. And those coiled mitochondria then can produce massive amounts of ATP to turn the motor. And remember, as we talked about, as that motor turns, it rotates in a whip-like fashion, that elongated tail region, not drawn to scale, or what is known as the locomotion region. This, as we know, is an organelle, known as the flagella, or flagellum, singular. That, again, gets turned in a whip-like fashion And that is what propels the sperm through space. And again, if you don't like my picture, here's the pretty picture from the textbook. Again, we can see all those mitochondria wrapping around the midpiece, wrapping around that motor region, that head with that acrosomial cap, and that elongated flagellum, that organelle, that is going to be that whip-like structure that moves along. Remember, as we talked about, this process is controlled by the nurse cells, who don't only release the chemical stimulants that cause this process, but as all that cytoplasm is shed, remember, it is them that are going to then reabsorb those nutrients and give it to the new immature cells as they're dividing and maturing, and they continue to recycle those resources. All right. As I mentioned, this entire process of getting from stem cell to sperm-shaped cell takes about 70 days, about 64 to 72 days. But remember, as I also mentioned, they are not capable of fertilizing an egg yet. They're still immature. So they need to mature. However, that maturation takes place outside of the testes. And that takes place in the tubular structure known as the epididymis. In the epididymis, it takes about 20 days for the sperm to mature. So if you think about it, to go from stem cell to cell capable of fertilizing an egg takes about 90 days, 3 months. You thought the lines at Disneyland were long. Imagine waiting 3 months for a 30-second ride. All right. And as I mentioned, our sperm has those 3 regions: the head or genetic region with the DNA and that acrosomial cap, the midpiece or metabolic region that contains all the mitochondria spiraled around, and the tail, the locomotor region, that flagellum, that organelle that propels the cell through space. And here's a pretty picture that shows this all. And notice the nice thing about this, again, this is an illustration, so it makes it super simple. But as I mentioned, one of the primary ways we're going to be able to identify these cells is by their locations. The cells that are right on the border of the seminiferous tubule are the spermatogoniums. The cells that have migrated away from the border and are by far the largest of the cells are the primary spermatocytes. As we move towards the lumen, that one primary spermatocyte has divided into 2 much smaller secondary spermatocytes, which then divide into even smaller, more densely packed early spermatids. Those early spermatids then elongate, form their flagella, becoming the late spermatids, which are ultimately released into the lumen of our seminiferous tubule. Color haven't I used? Brown. As individual cells. And notice your book even uses the term spermatozoa, but remember that these are still immature. And if we can do this on an illustration, can we do it on an actual light microscopy slide?
Yes.
Yeah, absolutely. Which, if I was really clever, would be the next slide. Nope. So instead, we'll go here. Notice this is a fairly low magnification, so we're not going to do too much identifying of the things on here, but there's still some things we can see. Obviously, these cells out here, actually that red doesn't show up very good, so let's just make a big fat arrow. How would you identify this cell here? Or these cells are even better. Identify those cells.
Interstitial.
Yeah, exactly. Those are the interstitial cell. What's their function?
Don't they produce testosterone and like the hormones?
Perfect, absolutely. What if instead I took this and moved it here? Identify this cell. Is it tricky because it's not in contact? Again, a little bit of an artifact. What if I put it here instead? See, I don't like it here because it's a little bit confusing. I actually liked that it was disconnected, but what if I did it there instead? What would you say there? There you go. Well, careful. It's not MYE, right? That myeloid. There you go. Myeloid cell. Exactly. Myeloid sounds like myelin. Myeloid cells. Excellent. This is that smooth muscle layer. Notice, obviously, there's been some damage to this, some separation, or really dehydration of the tissue that has occurred. And clearly, again, even at a low magnification, identify the organelles that are making this lumen look so dirty. Yeah, this one's even better. Identify those organelles. It doesn't show up well at all.
Are those the spermatozoa?
But I asked for the organelles. As I say many times in this class, people lose points not because they don't know the information, but because they don't read the question carefully. Yes, if I had said cells, spermatozoa would be acceptable, or late spermatids would be acceptable. But what I'm actually seeing are the organelles, the long tail-like structures. What are those?
Flagella.
Yeah, flagella. Absolutely, those are the flagella. Excellent. Now, if we increase the magnification, we can see things a little bit more clearly. Notice again, I like that one even better. Here we can see again our myeloid cells along the surface. We can see there's a nice example of one there. We can see the cells, and again, here we can see the flagella in much more detail. But let's look at this one. I like this one. Identify, let's make that even bigger. Identify the cell.
Is that a nurse cell?
Exactly. Excellent. Identify the cell.
Spermatogonium.
Exactly. The stem cells right next to the border. Notice how that compares to this cell. Identify that cell.
Primary spermatocyte.
Yeah. Identify this cell. Secondary spermatocyte. Excellent. Identify this cell.
Spermatid.
I'd give you that, although really I'm looking for early spermatid, but I'd like that. Notice again, there really isn't much difference in size between these two cells right here and right here. So how did we decide that this first arrow, here, let's change the color. How did we decide that the blue arrow was a secondary spermatocyte and the green arrow was an early spermatid. What allowed us to make that decision?
Because the secondary is right beneath the primary.
Yeah, location, right? Clearly, and we'll go ahead and use an even different color for this. Let's see if the brown shows up well. The primary, like I said, that primary spermatocyte is always so obvious because it's always so big and it's always easy to identify. So that's always a good landmark for a starting point. So yeah, the blue one is right next to it. So it clearly just finished dividing, whereas the green arrow is right next to the lumen. So even though it's really not that much smaller, its location is the dead giveaway to what it is. And then, of course, if we moved the green arrow to here, and I asked you to identify that cell, what cell is that? Yeah, late spermatid. Or again, I would accept the term spermatozoa or spermatozoon for the singular. But yeah, I like late spermatid because it reminds you that it is immature. So notice the illustration, obviously it's easy to see this, but even with the light microscopy, size and location are the big things that you're going to be looking for. And notice also, not only is the primary spermatocyte big in size, but it also is chock filled with chromosomes, right? It is 2NR. It is diploid and replicated. So it has the most genetic material inside. So not only is it huge, but they also have massive, massive nuclei, making those primary spermatocytes by far the easiest thing to see inside of these, with the exception of maybe the nurse cells with their elongated and distinctly different nuclei. Notice there are a couple more here as well, even one up here, but I thought this one was by far the most obvious. All right. Questions on that? All righty. So all of this occurs here inside of the seminiferous tubules, inside of one of these lobules, inside the testis. And so again, inside the seminiferous tubule, inside the seminiferous tubule is where we form the immature spermatozoa. But remember, we also talked about how this testis is part of a closed duct system. So if you notice, once this immature spermatozoa is produced, remember our nurse cells also produce fluid. This fluid helps to maintain the sperm. Oh, this doesn't need to all be in caps. But that fluid also produces fluid pressures. To move the sperm. And our myoid cells produce contractions to move the sperm. And they are going to pass out of the lobule. And they leave the lobule from the seminiferous tubules. And now that I've got all that explanation, let's actually just write this all out. From the seminiferous tubules, it leaves the lobule into a structure, as you can see here, that is known as the rete testis. and it gets there from the straight tubule. The straight tubule feeds into the rete testis, which is this kind of mesh-like, net-like structure located in the testis. It then leaves the testis via the efferent ductules. So again, if we were to draw this, we're starting in the seminiferous tubules, out the straight tubule, into the rete testis, from the rete testis out the efferent ductule, and that is how we leave the testis into the epididymis. Oops. And as we said, in the epididymis, this is where the sperm mature. Oh, what do we need to mature the sperm?
Hormone.
Yeah, what hormone?
Luteinizing.
Not a bad guess. Nope. Remember, it's actually the androgens. So indirectly, because luteinizing hormone causes the androgens to be produced. So how do the androgens get from the interstitial cells here into the epididymis? Well, if you remember, our nurse cells, not only do they produce fluid, but produce androgen binding proteins. And those androgen binding proteins enter the fluid that the nurse cells make. They bind our androgens and they carry them in the fluid to the epididymis. So here in the epididymis is where the sperm are going to mature. Let's switch here. I'm going to go ahead and cheat. Shorten my, so that we can. Because I want to be able to keep track of this. Seminiferous tubule into the straight tubule. From the straight tubule, what did it go into?
Retestes.
Excellent. From the rete testis into the efferent ductule. From the efferent ductule into what?
Epididymis.
Excellent. All right, so we'll leave that there and come back here. Again, it's hydrostatic pressure and those myoid cells that move it via the straight tubule into the rete testis. Again, not viable yet. Need to get into the epididymis via the efferent ductule for them to be matured. Notice the epididymis is not a part of the testis. It is a large comma-shaped tightly coiled tube, which if you stretched out from end to end would hurt a lot. And it has three main regions, a head, body, and tail. We also would describe it, oops, not the button I was looking to push, as superior, posterior, and lateral on the testis. Again, let's go singular. Once you have that male and you've observed his raphe, he has demonstrated the contraction of his cremaster muscle for you, gently palpate the testis. And if this was, for instance, my right testis, the epididymis, like a comma-shaped structure, is superior, is lateral, and is posterior. So it wraps around. So if you gently palpate the testis, you can feel this enlargement on the outer surface, like I said, lateral, posterior, and superior on the testis. And as we mentioned, it is within this structure that the spermatozoa are going to mature. Now, as I mentioned, there are 3 slightly different regions. They can actually be distinguished histologically, but I'm not going to hold you responsible for telling the 3 areas apart, and slightly different things happen in those 3 regions. But we will generalize and just focus on the 3 main functions of the epididymis. The first is to keep the sperm alive. It does this by monitoring and adjusting the composition of the fluid. Our epididymis is a very unique piece of tissue, something we haven't seen before. It is a ciliated pseudostratified columnar epithelial tissue, but it is non-motile. What does that mean?
Doesn't move itself.
It doesn't move, which if you think about it, doesn't make a whole heck of a lot of sense. Having cilia in there to move the sperm so they don't have to move themselves, wouldn't that make a tremendous amount of sense to have that in the epididymis?
Is it because the temperature? You said cilia is sensitive to cold.
That's an excellent point. It could be related to temperature. I think the other, and again, why is one of these things that we always have to, kind of guesstimate at. Temperature could influence their function as well, but also cilia would move them too fast. Remember, we want the sperm in here for 20 days or more to mature. So cilia might move them a little too quickly through there. But they do increase the surface area for adjustment of the fluid and maintaining the fluid. They need to store and mature the sperm, which, like we said, takes about 20 days. However, do we really want to keep every single sperm alive, store and mature every single sperm? If you've got a factory producing 500 million beanie babies every day, is every single one of them going to be perfect?
No.
No, I mean, those 8-year-old kids in China are impressive, but they're not that impressive. There are going to be mistakes. And that's true here as well. The epididymis also has the opportunity to recycle, break down, and remove dead or damaged or irregular spermatozoa. So a little bit of quality control can take place in here as well. Now, as I mentioned, it is a bit of a unique tissue, but it can be tricky when you first look at it. Identify the organ that we're looking at right now.
Seminiferous tubule.
True, but that's not the organ. That is the structure we're looking at. But what organ would this be?
Testis.
Testis. And you're absolutely right. This is the seminiferous tubule. Oh, there it is, up nice and close. Some more up nice and close. And even more seminiferous tubules. What's this? Well, based on what we're talking about, what do you think it is?
The epididymis.
Epididymis. right? Notice at first, these look somewhat familiar, similar to each other, right? However, there are some key characteristics that we can use to tell them apart. I know at first, when you see the cilia of our ciliated pseudostratified columnar, they may trick you into thinking those are the flagella of our spermatozoas that are, of our late spermatids that are forming. But if you look closely, notice all the nuclei are near the basal surface. Very, very few nuclei are located towards the lumen. Is that what you expect to see in the seminiferous tubules? Do you expect to see almost no nuclei near the lumen of a seminiferous tubule?
No.
No. Heck, if anything, you get more, right? You go from fewer cells to more cells as you go towards the lumen of our seminiferous tubule, right? If you think about it, in this space here, there is 3 cells. If I take that same box and put it up here, right, we easily got close to a dozen cells in there. So there are more cells, more nuclei as you go towards it. Whereas in the epididymis, there are far fewer cells that you see, far fewer nuclei that you see towards the lumen. Yes, we have those elongated cilia that can be tricky, but the lack of nuclei is one of the dead giveaways. The other dead giveaway is this clump of material right here. What the heck is this clump of material in the center of this tubule?
Sperm.
Sperm. Because think about what happens. We have here a tube that is not making sperm, but maintaining it. And then we want to put it on a slide. To put it on a slide, the first thing we do is we freeze the tissue. And a lot of tissue has water in it. So when we freeze it, it expands. Then we use a very sharp blade to slice off a very thin piece. And then we put it in all these different stains. And the stains tend to dehydrate it. And so notice when we dehydrate this tissue, all of the sperm clump together into this big independent mass in the center. I know these are slightly out of focus. These are just pictures I grabbed quickly off the interwebs. But here's one that looks much, much nicer. Notice you've got this huge clump of cells together, separate from the tubule, because this tubule is not making it. Notice when we go back to our seminiferous tubules, like this one, or this one, or all of these, do you see that same separation of the sperm in the center away from the tube that's containing it? No. So when you see nuclei throughout the layers, and again, that lack of separation in the lumen, that tells you you're in the seminiferous tubule. Here, we don't have that. The separation of the sperm, the lack of nuclei. And again, notice when we were looking at the seminiferous tubule, we were looking for small cells and then really big cells and then smaller cells and even smaller cells. Do you see that type of organization to the nuclei here? Is there anything here that looks like that big, massive primary spermatocyte?
No.
No. So while at first appearance, it can be tricky to tell these two things apart. If you pay close attention, you will easily be able to distinguish the epididymis from the seminiferous tubules histologically. All right. Questions on that? Excellent. From there, once our sperm spend their 20 days meandering through the epididymis, our tubule straightens out and leaves, forming the ductus deferens, or what is also known as the vas deferens. So you may have heard that term as well. And it is the vas deferens that then carries the sperm back into the abdominal pelvic cavity. It comes up through the spermatocord, and notice it goes over the pubic bone, basically right next to the pubic symphysis. So again, you guys have a lot of homework this weekend. You need to, Thanksgiving, you're getting together with the family. This is a perfect opportunity, right? To observe a raphe, observe the action of a cremaster muscle, palpate a testis, and then after you've done that, gently, again, gently, palpate the pubic symphysis of the male, and you can actually feel the spermatocords with that ductus deferens coming over the pubic bone and into the inguinal canal, where it then enters into the abdominal pelvic cavity and notice travels up and over the bladder. In fact, notice it hooks over the ureter coming down. So notice if we go back to our list, from the epididymis, we then go into the ductus deferens, or what is also known as the vas. I will accept both. Both are acceptable terms. While we're here, this is probably a good opportunity to talk about male birth control. What do we call the process of male birth control? I mean, other than saying no.
Contraception.
True. Okay, contraception. I was thinking of something a little bit more permanent than that.
Vasectomy.
A vasectomy, absolutely. The vas deferens, ductus deferens, is cut in a process known as a vasectomy, right? In ancient times, and by ancient times, I mean the 70s, when this first really started becoming popular, what they would do is they would actually go in, they typically make an incision underneath the penis in the scrotal sac, and they would actually put a clamp over the ductus deferens. Why would they do that?
Stop sperm from going towards the,
Exactly. But why a clamp? Exactly. Maximilian's got it. So it could be reversed, right? What happens if, for instance, you're married, you have your 2 kids, and then suddenly your wife dies? Or even worse, she gets old, right? You want to find a new, younger wife and have children with her. You want to be able to reverse that process, right? Of course, the problem with that is that the clips didn't always work there, 100% effectiveness. So they stopped doing that. So then they would just go in and cut the ductus deferens, and they found in a small portion of people it would grow back together. So now what they do is they actually cut out a section, and they actually cauterize the 2 ends of the ductus deferens, basically making it almost impossible to reverse. In fact, because most of these surgeries are done via HMOs, before you can have a vasectomy, there's a class you have to go to. I make light earlier, but they really do talk to you about what happens if your family dies in a horrible accident. What happens if your kid has some rare disease where you need to have a second kid so that they can get one of their lungs or something like this, right? They basically, because again, money and HMOs drive most of our medical procedures now. They'll pay to have it done, but they won't pay to reverse it. So they make you take a class to make sure this is really, really, really what you want, because it's pretty much a one-way street where they will go and they will do this, and that with over 99% effectiveness, this limits it. In very, very rare cases, it may grow back together, but again, it's very unlikely for it to happen. Obviously, there are two ductus deferenses, so depending on your doctor, they may make a single incision in the middle and hook both and bring it out, or two different incisions on the side to have it done. And it's an outpatient procedure. You know, the male goes in, has the procedure, puts a bag of frozen peas on his testicles for a day, and then he's back at work. Right? It's a weekend surgery. In fact, we'll talk about that in a second. We will get to that absolutely in just a second. I'll get to that in a second. I wanted to give one more anecdote. Several years ago, and I don't know if they still do it, there was in Minnesota, when the Golden Gophers were doing really well from a basketball standpoint, there was a urologist in Minnesota who did a March Madness special. On the Tuesday before the games began, he would do your surgery so that for the next 3 days, Wednesday, Thursday, Friday, or actually no, on Wednesday, because on Thursday, Friday would be the 2 days that you would be off from work. You could then be home recovering, watching all the games, and he actually threw in a free pizza. Now, to get to your point, Aravec, if you think about this from a plumbing standpoint, theoretically, is it possible that you could remove the cauterized portion, the scarred portion, attach these two tubes together and reconnect it? Yes. They will do it in rare instances because, like I said, for most people, it is a relatively painless process. But for some, the sperm production and the swelling of the sperm that can't be released from the testes can cause, in rare instances, very excruciating pain. And so to eliminate that pain, they will reconnect it. But what about for all those people that it isn't painful for? Well, then, as you mentioned, 2 years later, you could try to reconnect these tubes. But as Aravec pointed out, after a time, as you're continuing to produce sperm and they're continuing not to be able to leave the testis will actually stop producing sperm. So in as little as 2 years, your sperm production could go down to 0. So even if you did reconnect the tubes, you wouldn't be able to produce viable offspring because you wouldn't be able to produce enough sperm to be able to get someone pregnant. So it really is a one-way path, but it is much less dangerous, much less painful than a similar procedure in the females. So it is, for those who are serious about not having kids, it is a very, very reasonable and, like I said, relatively simple, basic procedure.
Is it like similar to milk production with, to females when they stop breastfeeding for a while, the milk just doesn't produce? Or it's totally different?
I mean, it's similar in the fact that production stops, but the mechanism from them would be totally different. Because here's the flip side of it. If the female gets pregnant again and produces those hormones again, they're going to produce milk again. Those hormones haven't gone away from the male, right? Basically what happened for the female is that hormone levels dropped, and as hormone levels dropped, milk production stopped. But once you bring those hormones back, they will continue to produce, they can start producing milk again. For a male, those hormones didn't go away. Basically, the testes just shut themselves off. So there's no way to turn them back on again. And yes, absolutely, Maximilian, you are correct. Because of the finality of the process, there are some men who will actually have their sperm frozen so that they do have the opportunity to have kids down the line if they want to. So yes, that is an option. You know, costs money, I assume, to do that. I don't know what that costs, but yes, that is something that they will do, absolutely. All right, excellent. Great questions. So again, ductus deferens over the pubic bone, through the inguinal canal, into the abdominal pelvic cavity, where, despite what your boyfriend may tell you, those sperm can be successfully stored for months, right? Sperm do not have to be released, right? They're not going to turn poisonous if they stay inside of you. Don't listen to what he's telling you. They can be stored there for months. Now, notice one other thing. Let's go back to that previous picture. As I mentioned, the ductus deferens enters the abdominal pelvic cavity over the top of the bladder to the backside. And notice on the backside, there is an enlargement of the ductus deferens known as the ampulla. And that ampulla is where those matured, ready to go, viable sperm are stored until it is time for them to be released. So a large portion of sperm is located there so that the massive amount that have to be released during ejaculation can occur. And we did the vasectomy, cutting that ductus deferens. Here we see that posterior view of the bladder, where again, our ductus deferens comes in, enlarges to form the ampulla. And let's finish the rest of the pathway. Notice the ampulla of the ductus deferens meets up with the duct of one of our glands, our seminal glands, or what are known as the seminal vesicles. And when these two glands come together, they form the ejaculatory ducts. And notice there are two ductus deferens, there are two ejaculatory ducts, and those two ejaculatory ducts feed into the one prosthetic urethra. Which of course then feeds into the membranous urethra, feeds into the spongy urethra, and is then out of the body. So if we're finishing off our pathway, we might as well put the ampulla which then feeds into the ejaculatory duct, which then feeds into the prosthetic urethra, which into the membranous, and then into finally the spongy or penile. And there you go. Those are, excuse me, the entire tubular structures, that entire closed pathway from sperm production to your stuffed animal. All right. Questions on that? All right. Excellent. How are we on time? You know what? I want to spend a little bit more time than rushing this in 20 minutes. So let's go ahead and stop here. This is a good stopping point, and we will pick up with the accessory organs, the glands, because there is a difference between semen and sperm. Sperm are obviously the fertilizing component, but semen contains more than just that. And rather than rushing through that, I think this is a good stopping point. So any questions on the rest of the ductwork structures we have identified going through on this? All righty. Well, then that is all that I have for you guys today. Again, study hard this week. We get a week off. Spend some time. Again, studying for this class is a perfect opportunity to hide from your family over the next few days. So feel free to spend time with them, but also feel free to study hard on this material. You've got a whole big week off. And yes, thank you. Actually, let me go ahead and do this. I'll